Cystic fibrosis. What is it? Cystic fibrosis is caused by the defect of a single gene, the cystic fibrosis transmembrane conductance regulator, or CFTR for short. It is autosomal recessive, meaning it occurs equally in males and females. The CFTR gene encodes for 1480 amino acid protein, normally functioning as a chloride channel apical membranes from epithelial cells, pumping chloride ions into mucus secretions to thin them out by drawing water in. Henceforth, people with cystic fibrosis develop an abnormal amount of excessively thick and sticky mucus within the lungs, airways and other physiological systems. More than 1900 possible mutations can occur in CFTR to cause cystic fibrosis, but approximately 70% of all patients with cystic fibrosis have the same defect, F508.
In 2015, there were 3,379 people with cystic fibrosis in Australia, of whom 52% were adults. The prevalence of the cystic fibrosis gene mutation varies in different ethnic populations, with the highest carrier frequency, 1 in 25, in people of Caucasian descent. The incidence of cystic fibrosis is rare in Australian Aboriginal, Torres Strait Islander and Maori people. Types of mutations that can occur to the CFTR gene can be classed into 5 categories: protein production mutations, protein processing mutations, gating mutations, conduction mutations, and insufficient protein mutations. Protein production mutations, which include nonsense and splice mutations, interfere with the production of the CFTR protein. Nonsense mutations cause a non-functional protein due to an early stop signal, while splice mutations also give a non-functional protein by incorrect removal or non-removal of regions of RNA. Protein processing mutations. The F508 mutation removes a single amino acid from the CFTR protein, causing it to lose its correct functional shape. The cell recognizes that the protein isn't the right shape and disposes of it. Moreover, missense mutations also fall in this category and occur when a change in DNA ladders causes an incorrect amino acid to be incorporated, leading to either a decreased quantity or function of the protein. Gating mutations. The CFTR protein is shaped like a tunnel with a gate. The cell can open the gate when chloride needs to flow through the channel, otherwise the gate stays closed. Gating mutations lock the gate in the closed position so that chloride cannot get through. Conduction mutations change the shape of the inside of the tunnel so that chloride cannot move through as easily as it should. And insufficient protein mutations cause a reduced amount of CFTR protein at the cell surface.
Now that we've reviewed the types of mutations in the CFTR gene, we can examine the impact of the primary physiological systems of most sufferers. It is important to know that other systems are impacted and will be briefly covered.
Firstly, the respiratory system. Patients suffering from cystic fibrosis are prone to infection from bacterial colonies, and the decrease in the amount of CFTR to bind with the bacteria leads to colonization of the airways. The clinical application of this would allow pathologists to test for bacterial cultures in patients to indicate that one does potentially have cystic fibrosis. As obstruction of the airway increases, this leads to expansion of alveoli, where air trapping occurs and over time causes a barrel-shaped chest, commonly in young adults. Further clinical application of this knowledge of increasing obstruction relates to decreases in FEV1, which is measured using spirometry. Further, the increased work of breathing that is characteristic to cystic fibrosis also serves as a clinical tool. Elements that determine increased work should be monitored, including breathing rate, grunting, nose flaring, retractions, and accessory muscle use. Typically, patients with cystic fibrosis have decreased levels of interleukin-10, a cytokine that has anti-inflammatory properties, especially in the lungs. The decreased levels predispose the patients to severe lung inflammation after infection. Sometimes, the pulmonary inflammation persists and becomes a chronic inflammatory condition, causing hypertrophy of the bronchial arteries and ultimately hemoptysis. Hemoptysis also serves as an excellent clinical tool as it is visually observable and indicates a patient may have the disease. Other systems that are affected include the gastrointestinal system via mucus blocking the pancreatic ducts, which alters the levels of enzymes that can be secreted, commonly causing issues with absorption. This is further linked to the endocrine system, as some cystic fibrosis sufferers can develop cystic fibrosis-related diabetes, due to obstruction of the pancreatic ducts that results in insulin deficiency. The reproductive system is also impacted, with most males being sterile due to a degraded vas deferens or malformation due to mucus blocking, and most women require more time to become pregnant due to mucus plugs in the oviduct and thicker cervical mucus to reduce sperm movement.
In its essence, cystic fibrosis is a disastrous disease that has widespread effects on multiple systems in the body. For such a small change in a single protein, the consequences are dire, and the changes to the livelihood of those affected are drastic.